This lecture is not difficult. I mean, it is just the types of the glial cells and their function. I don't think there is any concept. Like the last two lectures, there were concepts, concept like potassium leaky channels. This is a concept. Why potassium extra goes out. But here is only, we will quickly go through, you can read and let me know if you have problem. Because we are late already, and like maybe 15 minutes or 12 minutes, and the race is coming after me quickly. So, because you are tired, I will not take much time, inshallah. So, the topic is glia, and for glia are glial cells, neuroglial cells. It's the same word, neuroglia or glia, right? Actually, the neuron is the main cell in the brain. But there are two main cells, neuron and glial cells. Glia means connective tissue. You know connective? The connective tissue connects the different cells with each other. So they are labors. They connect two neurons with each other. If they will not connect, both neurons can go hither and thither. Oh, they will mixing, and it's not good how the function can occur. So they connect the neuron and other type of the tissues that called as connective tissue. So glial cells actually are also teachers of the neurons. You know why? In Sanatadiria we discussed. The reason is why. Huh? Yeah, you can take whatever. Remote, books, everything. See this? You take this. Leader will tell you. You can take books also. Okay, so these are teachers for the neuron. You know why? The reason is when neuron is born, very little child, very little, neuron cannot even attach with the surface. The fibroneck, there are proteins that attach the neuron with the inside the structures, but neuron cannot attach themselves. Neuron cannot properly move, and they cannot perform function. So glial cells, they tell them, oh boy, come, catch my hand, huh? And then the neuron, because they connect. The neuron do not know where to go and what to do. So glial cells tell them, you do this and don't do this. How to put tightly your feet, your parts to get strengthened. So it means the connective tissue not only helps, supports, but it also informs how to do activities. And number two, even this is physical help, but in fact they also give them money. 1,000 riyal, use it. Actually, they do the metabolism. Glial cells know how to do metabolism. They perform metabolism, they produce energy, and they give this energy to the neurons and neuron cells. This is a wonderful one. So glial cells help neuron physically. I told you physically they support how to walk, and metabolically. We talked in Sanatadi, if you remember. So did you understand this thing or not? And similarly in the physiology, normal physiology, growth, and all those activities. We will see slide by slide. But today, this lecture, there are no much concepts, just the names, types of the glial cells, like astrocytes and oligodendrocytes, types and their function. We will not go in anatomy, this is not anatomy. We will not go too much in physiology, not a little pathology diseases. So this lecture is easy, but you have to retain. This is the difficult thing. You have to memorize. Most of the names are memorization. So see, okay, we will see later, outline. Yeah, so see, these are the supporting cells. There is a connection between nerve cell and glial cell. This is most recent research. Glial cell and nerve cell connect and perform function. And the type astrocyte, microglia, and those ependymal cells, oligodendrocyte, Schwann cells. You remember Schwann cells? Yes. When it comes upon the axon, it gives the myelination. And you know myelination is very important for increasing the speed of the flow of the information or impulse. So similarly, central, there are two types of myelination, central and peripheral. Do you understand when I say central and peripheral? Yes. Central means brain spinal cord, and peripheral means anywhere in the body. And peripheral nervous system is not present in bones. It's present just without bones. But the central system is present in bones. Bony structure like the spinal canal or cranial nerve. So the peripheral nervous system is spread. If bones are there, then we will not be able to move. Huh? So the peripheral nervous system, no bone, boneless. And the central nervous system should be there. It should not move hither and thither. So, and the clinical significance. These are the topics. And I think we should keep, it's too big actually, but if I make it small, then it will be difficult. Anyways, yes. Now see, the name of the glia derives from it means glue. Glia is glue, attaching. But actually, this word adhesive is not a proper word. Glue means attaching. This sentence is not in your notes, okay? No problem, just for fun. If you like, you can write it. Otherwise, it's okay. Glue means they attach. Like you glue, put a glue, you know? Put a glue, attach the thing similarly. And so it means a glial cell attached with the neuron side. The neuron on the other side, they connect the neuron with each other. And they are the most of the cells, 90% of the brain cells are neuroglial cells. And they support neuron, I told you, both physically and metabolically. Can you explain, anyone, you, please? Physically means like how to attach on the surface, how to do work, how to get mature, what to do and what not to do. And metabolically means energy. And insulate, that's very important. Sometimes they do the insulate because those are connective tissue. And connective tissue might be the fatty. While neuron membrane is different, not completely fatty. So they insulate. And they guide the young neuron. Young means not the, like you young, the small. Young here means when they are born. Young is also used for the very little kids, you know, very, and they are not mature neurons to the proper thing. Promote health and growth. Okay, I think there's no difficulty. It's simple. And I missed one page probably. Did I? No. Let me know, please. No, this is simple types. Okay, this page is we will. And glia, this is the most important new concept. And people say gliotransmission, like glutamate, they serve as gliotransmitter. So gliotransmitter means when the neurotransmission occurs, postsynaptic cell is excited or inhibited, this gliotransmitter, they can change this activity. So it means they have some involvement, these glial cells. So interaction between glia neuron and the neurotransmitter released by nerve connect on glial cells. Okay? Those neurotransmitter can affect. And number 3, neurotransmitter can initiate calcium entry. You know that. Yeah. Calcium going to entry. Similarly to astrocytes, they change the membrane potential. Can be transmitted among a group of cells because of the presence of gap. You know, they jump from gap junction. So whatever you studied for the nerve cell, it can occur in the glial cells, but it occurs through electrical synapses. Okay? Gap junction means they jump from one side to other. Did you understand? It's simple. You read it. So should I repeat? The third one. Neurotransmitters can initiate action, calcium entry. This is easy. Into the cell. This cell is astrocyte. Okay? And changes in membrane potential, no problem. When it enters, calcium change after potential is changed, and potential can be, and this can be transmitted among the other astrocytes. I mean conduction, from one astrocyte to other. So it behaves like the nerve cell, though it is not the nerve cell, because of the breath. Now the reason is how it occurs, because they are not the nerve cell. They jump from one gap to other. I mean one side of the gap to other. Okay, because they are not the nerve cells. So how the transmission of this gliotransmitter occurs from one glia to other by gap junction, jumping. I think you remember from gap junction, electrical synapses are formed. Is it clear?
Yes.
Okay. Now, see, these are the two ideas. The left side is very established research. That is the nerve cell. Now, see, it interacts with glial cell. nerve cell and glial cell. Nerve means axon. And it influences, but in the presence of calcium. So calcium is very important. This is a recent discovery, particularly in the G proteins, G protein coupled receptors. It's not written in your notes. You can write that. GPCR means G protein coupled receptors. And there is another word somewhere, a protein of class G. I think we mentioned somewhere. Yeah, we mentioned. It's not written here. See, can you read this? The neuron, yeah, it's written. Neuronal activation of its type. GQ, GPCR. GQ means this is a type of G protein. GQ is a type of G protein. And G protein is very famous protein, having much function in the membrane, G coupled receptors. So this is established, but now this is not established yet, that whether the nerve cell it combined with the glial cell, what is the actual role of the calcium? I mean, in control and neuromodulation of calcium-dependent release of gliotransmitters. So this thing is still not finally known. This is the reference, but I think this is nothing. Just to remember a little. And again, the interaction. Substances released from glial cells alter the sensory post-receptive. You remember, same thing that happens in the nerves. And hence, neuroglial interaction, gliotransmitter, for example, ATP or glutamate, these serve as the gliotransmitters, neuromodulation that influence the synapse. So it means that synapses are also influenced by glial neurotransmitters, gliotransmitters. So this is emerging new research, okay? Emerging, basically the glial cells are not the nerve cells. And before that, we used to say these are not excitable cells. When the things are changing, where I told you wherever is the ion channel will serve as the excitable cells, okay? So, but I did not go more because you do not study books. So this is the idea. And after the release into synapse, glutamate, one is the acetyl, in neuromuscular junction. But in synapses, if the neurotransmitters release this glutamate, it is transported into astrocytes also. Not only to the postsynaptic cell, but also astrocytes, which convert it into glutamine. And you remember, glutamine is recycled in the nerve cell. This is very important. You know that? Particularly gamma-aminobutyric acid. This is metabolized only in brain, not in the whole body. So Krebs cycle, glycolytic cycle all occurs in whole body, blood. But just one more, things like glutamate, conversion glutamine, or GABA, these are done by the brain. So why brain makes it? Because the balance of nitrogen, it is under the control of the brain, and this also relate to balance of hydrogen. You say oxygen is important after life. In Sanatadiri, I told you after life oxygen is important. But actually, new research shows that hydrogen balance is also very important. So hydrogen balance through the glutamate by the nitrogen, I don't go in details, is done by brain. So this thing is also there. So glutamate is converted to glutamine, and there is a role of glial cells. This I am saying. There is a role of regulation of glial cells. So research is going on. This is just a few steps. Scientists are continuing. Is there any question? Yes, sorry.
Glial cell, is it excited?
Glial cell?
Is it excited?
No, it is not excited. According to definition, according to your level, glial cells are connective tissue cell, and that connective tissue cells are not excitable. We say body has two types of cell, excitable and non-excitable. Excitable cell, if you stimulate electrically, you was not present in the last class. 8 to 9? You was? Okay, good. So if you excite electrically, it gives response excitable cell. If you excite electrically, no response, non-excitable. But I'm saying that with the time, we are understanding that there are many structures that contains the excitable molecules. And what are those excitable molecules? Ion channels. So astrocytes have excitable molecules. So it means there is some sort of excitability in the astrocyte also. They can work just like, to some extent, like neuron. For example, information goes by gaps from one astrocyte to other. What it shows? Definitely there will be ion channels, other things. Otherwise, it's not possible. How the information is made? I mean, conveyance is by gap, but making information needs ion channel. Understand? But I don't go more, otherwise you will get confused. The astrocytes and all those glial cells, they are non-excitable cells. Okay? They are non-excitable. The neuron and muscle cell and all sensory cells, those are the excitable. Is it clear? But I also talk about the concept of excitability at the molecular level. After the okay, you got it. So glutamate is changed to glutamine, and here the glial cells are there. People are doing work, and it will take few years or more. Then things will be clear. Maybe at the end we see that glia and neuron, they work together to perform some physiologic function. And we are explaining only at this time by the nerve cells. With the time, maybe no, interaction of nerve cell and glia cell, particularly immunity. You know, immune systems, people say it relates to blood and hematology, but actually it's the area of neuroscience. Because the glial cells are actual cells related to immune process. So things are related. With the time, we are getting the things. Okay, this is okay. I'm sorry, we do not have time. I talk only what is written. I don't go away. Astrocyte. Now see the astrocyte, the shape is, they are huge, large number. Number one, they are large numbers. They have different shapes, versatile. Versatility means different types of the astrocytes and branch cells. Okay. They are irregular shape, roughly star shape. Okay, you can read. I think there is no anything. They cling. Cling mean attached with the neuron. Now tell me, definitely there is something, interaction between the astrocyte and neuron. Though we don't know much. But if it comes and cling, what it means? They are talking with each other. There is role of the clinging. So they cling to the neuron, and there's synaptic ending also. You know the axon at the end is made into hundreds of the endings. They also cling over there. And over the capillaries also. So all these things show the functions of the glia. We will talk about the functions later. After that, now the support and brace. Brace means tightly, wrap tightly. Okay, they fasten tightly. I think this word is not written. You can write. Brace is written in your notes. Okay, is it written? No. But you can write in bracket to fasten tightly. So they support and brace them. Now this is a big function. I mean, if they do not tighten the neuron with each other, how neuron will talk? When I say talk with each other, means communicate. Information, one neuron gives to other that back. So how they will communicate? I mean, they will be moving like this, but the strong support in between and connection. Blood is also connective tissue. You know that? Bone is also connective tissue. Why? Because they make continuity. They connect the structures with blood, connects the body, different parts of the body. Bone connects the different, so this is connective tissue, okay? And they guide, okay, the young neuron, where to go, where not to go, where to migrate, how to migrate, who teaches glia. They control the chemical environment by buffering. Potassium, this is easy, and recapturing the transmitters. They're believed to be essential part of the blood-brain. Now see, this is another big function. You know what is blood-brain barrier?
Yes. Yes.
All the things cannot go into the brain. Big boss. Most of the things remain in the body. But some of the important things have to go into the brain for function. And in fact, some viruses also go. Very, very dangerous virus. But actually most of the things are not allowed to go. And these perform function. The glial cell perform function. Which things should go, which things should not go. So this is partly regulated by the glial cells. And I was studying some of the drugs. Even some drugs can go into the brain. Some of them cannot. Recent technology in medicine, when you will go at higher level in your specialization, you will study that sometimes if you have to transport a drug as a clinician, and you give drug to the patient, maybe it will not go to your destination, manzil maqsood. destination. So nowadays what they do, they prepare a little lipid membranes, because lipid membrane can go everywhere. In our body also, small lipid membrane is also lipid. So they make lipid, we say liposome. But nowadays we make nanoliposomes. Very small, very, very small. And we put a little drug in it, and then give to the patient. We know when it will be broken, how long it takes to be broken down. So it will reach there, it will go in brain or not go. It should be broken before going to brain. This is very wise, we're full of wisdom technology. Sometimes we cover it and wrap with the drug, some substances, to make it different. Sometimes we make one layer, like insulin, diabetic patient layer, 3 types of insulin, one, another one, and third one. So maybe after few hours, one is broken down. After other few hours, the other is broken down. After the third one is broken down. So this is the reason the blood level is controlled in 24 hours. 6, 7 hours. Because we eat food. We eat food. We do not eat food always. Anybody eat food always? Okay, we can imagine. But we eat food one time, then second and third. So in between, how glucose will be regulated? So that insulin is available. So there are different type of the nanoliposomes. So similarly, the people are doing work how to understand the drug interaction in perspective of the glia in the role of blood-brain barrier, how drugs go. So it means glial structures are very important in understanding how drugs go into the brain, cross blood-brain barrier. I don't go more. I think this is more than needed. Okay, leave it, throw it out. All right. So I think sometimes you just listen. Listening is enough. Maybe after 10 years, you are a big researcher. This thought can help you. Really? And particularly, in the last lecture, I told you a new microscope has been invented. Its name is scanning tunneling microscope. You read it, okay? Because my purpose mainly is to make you very wise and full of wisdom. To be very frank, not only you get the marks and become doctor. No, more than that. Because in the coming time, you become very higher level. My purpose is a little different. So I usually put into your mind and you do not know. Then I go, but it remains there. Now I put in your mind that in the recent technology, coming time, you have to, when you become a medical doctor, a specialist, you keep eye on this microscope and try to understand the pathogenesis and pathophysiology, how diseases occur with the help of the scanning tunneling microscope. Actually, scanning tunneling technique was the original technique. Someone got Nobel Prize. But recently, a very nice microscope has been made, and its name is atomic force microscope. What is the name?
Atomic force.
AFM. We usually say AFM. We do not have, I don't know, in Saudi Arabia, we do not have. Electrons microscope we have, but this is more expensive. So AFM is, when you go home, you read in Wikipedia somewhere, you write atomic force microscope. So this will be very helpful, and it is derived from scanning tunneling, the original technique physicists people discovered, scanning tunneling. They use, but we people don't understand. It's too much deep, because they study atoms. We don't study atoms. We are medical people. We need to understand the processes and how they occur in pathogenesis. So for us, another technique, atomic force. What is the name? Atomic force. So when you go home, anytime you get time, please read what is, because then you can apply in your work and in your areas, future areas, when you will become the student, takhassus, you will get takhassus, then it will help. So that is the most recent discovery. It's very important for us to. The role of the glial cells and central nervous system. See, it means nerve cells develop and they help glial cells. And not only development, but regeneration. You understand regeneration? Sometimes nerve cells degenerate. And this is a very unfortunate that all cells of our body, we understand, they become again 1 to 2, 2 to 4, but nerves are dead. Now these, with the help of these glial cells, maybe in future one of you or many of you can discover a new thing. Oh no, nerve cells can also be regenerated. And this will be one of the highest fayda for us, for humanity, if you can regenerate the neuron. So maybe through glial cells. because glia are there, and if we interact the glia with neuron, who knows, maybe the neurons are regenerated. Actually, some of the work is done. And the part of the glial limiting membrane, I mean, the membranes of the glia, they protect the neuronal functions. So all these things show the importance of glia, the role of gliosis. Gliosis is normal and abnormal. This is a disordered process when some infection occurs and the manifestation is gliosis. But it can be good, like sometimes inflammation is good. When inflammation occurs, the recovery occurs. Similarly, it's written here, it's not in your notes, but you can see dictionary. Gliosis is a non-spaghetic changes of glial response to damage to nervous system, central nervous system, okay? And it can be due in health or physiology or pathophysiology, okay? So, and scar formation. So it means when the scar is formed, interesting. So the glial cell might be involved. So it means the scar or gliosis can be controlled if we know the further the function of the glial cells. These are the functions. Okay, astrocyte. And now the second is, we were talking about astrocyte, right? The haphazard, diverse shapes, numerous, most numerous. Now these are oligodendral. You remember the Schwann cells and membrane wrapping with the, this is something. It means few, and they're small, few short. Okay, they wrap the central, not the peripheral, central nervous system, myelination, and therefore they're responsible for the huge speed. When myelination is there, node-to-node jumping, saltatory conduction. See how much important is the glial cell. If glial cell is not there, I cannot talk fast because I have a type of the axons that are myelinated. And with the help of that, I'm talking too much fast and movement you are seeing and you are thinking and you are responding. All that is because of the myelinated oligos. So this is very important. Yes. In this lecture, as I told you, just the names and functions. Now, ependymal cells. This is the third type of the glial cells. They are ciliated epithelium and they line the central cavities of, again, central nervous system. Only brains might have. And they help circulate the CSF. They're covered by villi and have central cluster of lungs. I think you have to remember, there is no concept. Two things you have to remember. And they are ciliated. In this whole slide, we know that a type of the glial cell that is ciliated, I mean the cuboid or columnar, and hence it lines the central system and performs the